Dear all, I am Dr. P. Saujanya, Professor and HOD Pharmaceutical Biotechnology from Vignan Pharmacy College. I welcome you all to the lecture on identification of bacteria by staining technique. The bacteria are microscopic organisms and they are colorless. So, these bacteria cannot be visualized properly under the microscope. In order to enhance the visibility of microorganisms and as well as to study the structural features and shape of the organisms, these staining techniques was developed. And the addition of these stains becomes more visible of the organism. And this means that the structures have to be contrasted from their environment so that they can be seen easily. What is a stain? Stain is a dye used to color the living or dead organisms. Here, different stains has been used to identify different types of organisms. And these are the various types of stains: acidic stain or acidic dye, basic dye, and neutral dye. Acidic dye are negatively charged acid radicals, and these negatively charged dyes they impart color to the organisms, and the examples of these acidic dyes are eosin red, acid fusion, malachite green, nigrosin dye, Indian ink. Basic dyes are positively charged and they have basic radicals combines with negatively charged particles in cytoplasm and gives color. The basic dyes are positively charged. So as they are positively charged, they stains the negatively charged molecules present in the cells. So here you can see they are staining the cytoplasm. The cytoplasm contains phospholipid bilayer. So the phospholipid bilayer contains phosphate ion, hence it gives negative charge to the cytoplasmic membrane. So the positively charged dyes will give color to the negatively charged organisms or negatively charged organelles of the cells. Examples of these basic dyes are hematoxylin, methylene blue, crystal violet, gentian violet. All these are basic dyes. Next, neutral dyes. Neutral dyes are they are both positively and negatively charged and imparts different colors to different components. They are both positively and negatively charged dyes, and they give different colors to the different cell components. And examples for neutral dyes are Giemsa stain, Leishman stain, Wright stain, etc. So positive staining. The staining techniques are of two types, positive staining and negative staining. Positive staining means which impart color to the cell components. Negative staining is the cell components which cannot take up the dye. So the negative staining will create the dark background. In the midst of the dark background, the lighter bodies of organisms are seen. So the positive staining is where the actual cells are themselves colored and appear in clear background. So the positive staining techniques are simple staining and differential staining. The simple staining includes a single stain. It is also known as monochrome staining. It uses single stain. And this gives colors to the bacteria and all the cell components. So the simple staining is used to study the shape, size, and arrangement of the bacteria. This staining will not able to differentiate different types of bacteria. And examples of simple staining is Loeffler's methylene blue, polychrome methylene blue, diluted carbol fusion. All these dyes are used in simple staining. Differential staining is which is used to bring out the differential characteristics of the organisms, and this includes one or more dyes. And if two dyes has been used in differential staining, the first dye is known as primary dye, and the second dye is known as secondary dye or secondary staining. And examples of these differential staining techniques are Grams staining, acid fast staining, and special staining techniques. Negative staining. Negative staining where the cells remain clear, that is uncolored, and the background is colored. So here the background is created, and in the midst of the dark background, the lighter bodies of the cells has been visualized. Examples of these negative staining are Indian ink and nigrosindyne. These two stains are used in negative staining. And before the staining has been done, the smear has to be prepared. What is the smear and how the smear has been prepared? The smear is distribution of bacterial cells on a slide for a purpose of viewing them under the microscope. That is distribution of a bacterial cells or spreading of a bacterial cells. If you take a loopful of suspension, then it contains multiple organisms. If you place a drop on a slide, it cannot differentiate the organisms, and as it contains a group of cells, you cannot visualize properly under the microscope. That is why a smear has been prepared. Smear is nothing but spreading the organism on microscopic slide with the help of a inoculation loop or inoculation needle. Okay, so this spreading causes distribution of organism on the microscopic slide, so that it can able to visualize individual organisms on the slide under the microscope. So here from any solid media or from the liquid media, the smear can be prepared. If it is a solid medium, first you need to take a drop of water, that is a sterile water. Then with the help of an inoculation needle, mix the bacterial culture so that it mixes up properly, and then spread the organism on microscopic slide. Here you can see the spreading of organism along with the water drops. So from if it is from a liquid medium, then uh, the inoculation loop has been taken. The suspension is taken with inoculation loop, and the suspension has been spread over the surface of the glass slide, and that is the preparation of smear. So after preparing the smear, either from taking a solid medium or from a liquid medium, then it has to be air dried, and then heat fixation. Heat fixation is fixing this organism to the solid surface, that is glass slide. Now this smear is ready for the staining purpose. So whatever the smear has been prepared, after air drying, it should be fixed by heat fixation process or methanol fixation. Heat fixation means it is the smear has been passed over the Bunsen burner flame so that it causes death of the organism, and as well as it causes fixing of the organism to the microscopic slide. Once it has been heat fixed, then it should be allowed to cool before the stain has been added. Why? Because when the slide is in hot condition, if you add the stain, then the stain sticks to the glass slide. It should not be like that. So it should be cooled before it is used for staining purpose. Methanol fixation. Methanol fixation here, after the air dry, then the methanol has been added to the smear, and it is allowed to stay for 1 minute. And meanwhile, the methanol has been evaporated, and it causes fixing of the organism to the microscopic slide. And these are the methods of staining. The first one is simple staining, also known as monochrome staining. So as I told you earlier, simple staining includes only single dye. So before the staining process has been done, first you need a culture, a suspension of bacterial culture, and a clear or clean glass slide. So this is a clean glass slide, and using an inoculation loop, the bacterial suspension has been taken, and it is spreaded on the surface of the glass slide, and then it is air dried. Then the methanol has been added. So here The process is methanol fixation. If it is not methanol, you can also heat fix. So heat fix the smear. Then flood the smear with a dye. What are the dyes used? Loeffler's methylene blue or crystal violet, saffronin. Any of the stain you can use, but only a single stain. Flood the smear with the stain. Then, after allowing it to 1 minute, then the stain will penetrate into the cell wall of the bacteria. Then you have to rinse the excess of the stain with water by washing with water. Then blot to dry and observe under the microscope. When you observe under the microscope at 100x in oil immersion objective, then you will identify the shape and arrangement of the bacteria. Here you can see the arrangement of the bacteria. These are the cocci, which has been formed as a bunches. And these are the bacilli, rod-shaped bacteria. So like this, you can identify the presence of bacteria by simple staining technique. Next one, Grams staining. Grams staining is differential staining to differentiate gram positive and gram negative bacteria. So in the gram staining, first the crystal violet dye has been added to the smear. And before the staining has been done, first you need to prepare the smear and then heat fixation or methanol fixation, then stain with crystal violet. And then a mordant has been added. In the Grams staining, the mordant is iodine. Once you add the iodine, this enhances the penetration of this crystal violet dye into the cell wall. That is the function of mordant. Mordant means enhancement of penetration of dye into the cell wall. And some of the bacteria are able to retain the primary stain, that is crystal violet, and some will decolorize after washing with alcohol. So let us see what are these cells which retain the primary stain and which will lose the primary stain after decolorizing with alcohol. So the cell wall of gram-positive bacteria have a thick layer of protein sugar complex, that is peptidoglycan. The gram-positive contains thick layer of peptidoglycan and a gram-negative contains thin layer of peptidoglycan. But the gram-positive contains as thick layer of peptidoglycan and the lipid content is low. Whereas gram-negative contains thin layer of peptidoglycan and thick layer of lipid content. So after staining with the crystal violet and treating with the mordant iodine, then the decolorizer, that is alcohol, has been added. This alcohol dissolves all the lipid content in the cell wall. As the gram positive contains thin layer of lipid content, then very few amount of dye or stain will be washed away. But a gram negative cells contains thick layer of lipid layer and thin layer of peptidoglycan. So as it contains thick layer of lipid, so all the stain that has been present in the cell wall has been washed away in gram negative cells. So the ethanol removes the crystal violet iodine complex that is bound to the peptidoglycan layer of gram positive. So and it appears as blue or purple. So the whatever the alcohol treatment has been done, it will not able to remove the gram positive bacterial cell wall stain because as it contains thick layer of peptidoglycan and thin layer of peptidoglycan, thin layer of lipid, then it will not be washed away. Whereas gram negative cells contain thin layer of peptidoglycan and thick layer of lipid. So the lipid content, once it has been dissolved, it eliminates all the dye that has been penetrated in the cell wall. So the gram negative bacteria loses the primary stain. So what happens once it loses the primary stain, it is ready to take up the secondary stain, that is saffronin. So the gram negative bacteria appear as red or pink in color. Gram positive bacteria appears as purple color. Okay, let us see the procedure of gram staining. So first you need to take a clean glass slide and then prepare the smear and heat fix the smear. Then gently flood the smear with crystal violet dye. So after addition of crystal violet dye, allow it to stand for 1 minute. Then tilt the slide slightly so that it will the crystal violet dye will be spreaded on in the entire smear. Then it has to be washed with water. Then gently flood the smear with grams iodine. So this grams iodine acts as mordant that enhances penetration of the dye into the cell wall. So after addition of the iodine, allow you to stay for 1 minute. Then decolorize with 95% ethanol or acetone. So this decolorization step is very important to differentiate gram positive and gram negative bacteria. So after treating with ethanol or acetone, the gram negative bacteria loses the primary stain, whereas gram positive bacteria retains the primary stain. So after treating with ethanol, then rinse with water and flood the smear with saffron in and allow it to stay for 45 minutes. This 45 seconds. This saffronin is a secondary dye or a counter stain. So as the gram positive bacteria retained the primary stain, it will not take up the secondary stain saffronin. The gram negative bacteria, as they lost the primary stain, it takes the secondary stain saffronin. Hence, the gram positive bacteria appear as purple, gram negative appear as pink color. So after staining with the counter stain, rinse thoroughly with water, blot to dry, and then observe under the light microscope under oil immersion 100x. There you can differentiate gram positive and gram negative bacteria. So these are the simplified steps. So prepare the smear and after heat fixation, add the crystal violet dye. So both gram positive and gram negative will take up the primary stain here. Then add iodine. Iodine enhances the penetration of the dye. Then treat with decolorizer, that is ethanol or acetone. So here what happens? In this picture you can see all the gram negative bacteria lost the primary stain because of thin layer of peptidoglycan and thick layer of lipid content. The lipid content dissolved and so that it eliminated the primary stain crystal violet. So after this decolorization treatment, then the smear is treated with a counter stain saffronin. So as the gram positive bacteria, as it retained the primary stain, it will not take up the secondary stain saffronin. But the gram negative bacteria, as they lost the primary stain, then they will readily take up the secondary stain. So here gram positive appear as purple and gram negative bacteria appear as pink color. So these are the gram positive cocci and gram negative bacilli. Cocci are spherical shape or round shaped bacteria. Bacilli are rod shaped bacteria. And the next staining technique is acid fast staining. Acid fast staining, also known as Gill Nielsen's staining. So it was described by Franz Gill and Frederick Nielsen. Both described the acid fast staining. And this staining technique is used to identify mycobacterial cells. So these mycobacterial cells in their cell wall, they contain mycolic acid. They doesn't have peptidoglycan. They have mycolic acid. The mycolic acids are beta hydroxy carboxylic acids, and they will have a chain length up to 90 carbon atoms. So the acid fastness is related to the carbon chain length of mycolic acid. So the acid fastness exhibited by mycobacterial cells is depends on the length of this carbon chain present in the mycolic acid of mycobacterial cells. And this staining technique divides the bacteria into two groups. They are acid fast bacilli and non-acid fast bacilli. Acid fast bacilli are Mycobacterium species, non-acid fast bacilli are non-mycobacterium species. And this is the procedure for acid fast staining. The smear has been prepared and then add a primary stain, carbonyl fusion. Okay, this after addition of this carbonyl fusion, boil the smear. So this boiling enhances the penetration of the dye. So in the earlier gram staining, we have used iodine for as mordant for enhancing the penetration of dye. But here, as this mycobacterium cell wall uh, is a thick layer, so it needs a harsh treatment. So that is why the smear has been heated for the enhancement of penetration of dye into the cell wall. Then decolorize the decolorize the smear with the addition of the acid alcohol contains sulfuric acid and in alcohol solution. This acid alcohol, it dissolves the primary stain of non-acid fast bacilli. So here the spherical shaped, they lost their primary stain because they are non-acid fast bacilli. But here what are the rod shaped bacilli you are seeing? They are acid fast bacilli. So they retained this primary stain even after decolorizing with acid alcohol. Then the smear is treated with a secondary stain. Secondary stain is methylene blue, also known as counter stain. So as the non-acid fast bacilli lost the primary stain, they will take up the secondary stain, methylene blue, and appears as blue color. And the acid fast bacilli, as they retained the primary stain carbonyl fusion, they appear as pink color. Okay. Next, endospore staining. Endospore staining is for staining of endospore bacteria. These endospores, they cannot be easily stained by normal staining process. It also needs harsher treatment as that of acid fast bacilli. And the method used for endospore staining is Schaffer-Fulton method. So in this method, the smear has been prepared and the smear is air dried and then heat fixed. Then the primary stain malachite green is added. So malachite green is the stain which is used to stain endospores. Then warm the smear so that it enhances the penetration of this malachite green into the endospores. Then after this warming or keeping the smear to the steam should not evaporate the stain. That is, malachite green should not be evaporated, but it should enhances the penetration of the dye into the cell wall. Then remove the slide from the hot plate and then cool it and gently rinse the slide with water and then add saffronin. Saffronin is counter stain or secondary stain. So whatever the bacteria which doesn't have endospore, they will lose the they will not take up this malachite green stain. And as they cannot take up this malachite green, they will take up the saffronin dye. And whereas the endospore staining, they will take up the malachite green stain. So hence they will not take the saffronin secondary stain. So the endospore bacteria appear as green color, non-endospore bacteria appear as pink color as they are stained with saffronin. So here you can see the endospore staining process. Prepare the smear, then treat the smear, air dry the smear, and then heat fixation. After that, apply the or flood the smear with endospore stain, that is malachite green. So after addition of the malachite green, both non-endospore and endospore forming bacteria will take up the dye. Then Uh, the smear has been boiled for few minutes for enhancement of penetration of the dye into the cell wall. Then it is washed with water or decolorized. So the non-endospore forming bacteria will loses the primary stain malachite green, whereas endospore forming bacteria retains the primary stain. So after decolorizing with water, then the smear has been treated with safranin, counter stain safranin. So here the endospore forming bacteria, they will not take up the secondary stain because they are already stained with malachite green and they appear as green color. Non-endospore forming bacteria appear as pink color. Next, capsule staining or negative staining. So capsule, it is a cell component which is externally covered to the cell, and this capsule cannot take up the dye. So to identify or to differentiate capsulated and non-capsulated cells, this negative staining has been used. Negative staining is, it is creating the dark background. Positive staining is the cell components has been stained so that they will appear in different colors. So here in the negative staining or capsule staining, the smear of the capsule has been prepared in such a way that the bacterial suspension of one drop has been taken and it is mixed with the stain, nigrosin dye or Indian ink. Either one is used in capsule staining or negative staining. So here the smear has been prepared by spreading with the other clean glass slide. So place near the drop of the smear has been prepared. That is that containing a drop of bacterial suspension and the dye, that is nigrosin dye or Indian ink. Then using the second slide, spread the suspension towards the other end so that the culture has been thinned out. So like this, you have to spread the second slide from one end to the other end. And this bacterial suspension along with the dye has been spread evenly so that it causes thinning of the suspension. And thereby, once it has been dried, if you observe under the microscope, the capsulated cells has been appear as lighter bodies in the midst of the dark background. Here you can see the capsulated cells has been appear like this, lighter bodies in the midst of the dark background. These are the capsulated cells. Next, flagella staining. Flagella staining is to identify the presence of the flagella and the arrangement of the flagella. So flagella are thin hair-like structure made up of a protein known as flagellin. So the size of the flagella ranges from 20 to 200 micrometer in length, and it is very thin structure which cannot be visualized with light microscope, and it can be visualized by electron microscope. So here this flagella staining, it needs a dye which enhances the thickness of the flagella. It needs a dye which enhances the thickness of the flagella. And the flagella are generally present in rod-shaped bacteria, but in few cocci, the flagella is present. So here, the leaf sense stain is used to enhance the thickness of the flagella. The leaf sense stain includes tannic acid and basic fusion, which enhances the thickness of the flagella. So this is the procedure for flagella staining. The method used is leaf sense method. Take a drop of bacterial suspension and place on a clean glass slide and kept in slant position. And now it is mixed with uh, leaf sense stain. It is mixed with leaf sense stain. Leaf sense stain contains tannic acid and basic fusion till A thin film of shiny surface appears. It should be mixed with leaves and stain. Then wash the smear and treat with 1% methylene blue. 1% methylene blue. So after treating with methylene blue, wash thoroughly. The excess stain has been washed off. And then once you observe under the microscope, the flagella of the bacteria appear as red color and the bacteria appear as blue color. So here in these images, you can see the bacteria appears as blue color and the flagella appears as pink color. Okay. So the pink color is due to leaf sense stain and the blue color is due to methylene blue. Okay. So both has to be used and they will either of these stain will not stain both the bacterial cell and as well as the flagella. So two different stains has been used to stain flagella separately and the bacterial cell separately. This is flagella staining. And these are all the techniques used to identify the microorganisms by staining techniques. Thank you.